Hi there. In this video, I'll talk about protein trafficking within a cell. We already talked about organelles, and there are membrane-bound organelles where proteins have to somehow cross those membranes. So this is talking about how they find those membranes, how those protein products find the membranes, and then how they enter through those membranes into the inner parts of the organelle. And if they have multiple membranes, then they have even a more daunting task to get through more than one membrane. Here is a plant cell, and most of the proteins are encoded by genes in the nucleus in these plant cells. So there's expression here in the form of transcription of the genes in the nucleus. Then that mRNA exits the nucleus, winds up either in the cytosol or in the endoplasmic reticulum, and then it's translated into proteins in either the endoplasmic reticulum or the cytosol. And some of those proteins have to end up in the chloroplast, in the Golgi, at the cell membrane, in the mitochondrion, in the peroxisomes, in the vacuole. And the question is, how do they get there? How do those proteins wind up in those different compartments? And it's accomplished by a process known as protein trafficking within each one of those cells. And the situation is the same for animal cells and protist cells and all eukaryotic cells, as well as bacterial cells. Anytime a protein has to end up in a certain compartment, except the cytosol, there have to be certain signals on those proteins in order for chaperone proteins to carry those to the proper organelle, the proper membrane, so that they can do their job. So they don't just wind up out in the cytosol randomly and just happen to enter a mitochondrion or enter a Golgi or go to the plasma membrane. They're actually shuttled to those different regions. So the RNAs can wind up in two places, right? Cytosolic ribosomes and endoplasmic reticulum ribosomes. But as the RNAs exit the nucleus, they are immediately attached to ribosomes, and those ribosomes then start translation. If the first part of the protein emerging from the ribosome has a particular sequence on it, amino acid sequence now, it will attach to a particular protein, a specific protein, which will then attach it to a receptor on the endoplasmic reticulum membrane. If it does not have that particular amino acid signal on the end, called a signal sequence, it will end up being further translated in the cytosol. Here's a diagram of that process as the mRNA exits the nuclear pore on the nucleus. It's immediately attached to ribosomes, and then translation begins. And the amino end is the first to emerge on that protein, and if it has a signal peptide, then a protein will attach to that and shuttle it to one of two places. The first is to the endoplasmic reticulum, and that then attaches to a receptor on the ER membrane, and the protein is then threaded through into the ER lumen, and translation continues. If it has a signal there to remain in the ER, it will, either on the membrane or in the ER, or it may be sent to the Golgi complex and then become part of a secretory vesicle, part of a lysosome, or part of the plasma membrane. Alternatively, if it has a different signal peptide on the end or no signal peptide on the end, it will remain in the cytosol, not on the rough ER. If it has no signal peptide, it will be a cytosolic protein. That's where it will end up. If it's destined to go into the nucleus, like for RNA polymerase, DNA polymerase, etc., then it will be attached to a protein that will take it back through into the nucleus. If it's to wind up in the mitochondrion, it will have a different signal peptide, sometimes more than one signal peptide, to take it through one or two membranes to wind up in the matrix, on the mitochondrial inner membrane, etc. If it's to go to the chloroplast, it has different signal peptides that will take it through one, two, or sometimes three membranes all the way into the thylakoid membrane. And then if it goes to peroxisome, that will have a different signal peptide. Here's a schematic of that process. So the transcription is in the nucleus. That mRNA exits the nucleus. And then if it's destined for a rough ER ribosome, it will have a signal peptide on that protein, emerging protein, which will deliver it to the endoplasmic reticulum, and then it will either end up in the endoplasmic reticulum or passed on to the Golgi, where it will become part of a lysosome, a secretory vesicle, or part of the plasma membrane, a protein in the plasma membrane. If it has different signal peptides, it will end up being translated in the cytosol. And if there's no signal peptide at all, it finishes being a cytosolic protein. However, if there's a signal peptide that makes it destined for the nucleus, a protein will attach to that and take it to the nucleus. If it's destined for the mitochondrion, it will have a certain signal peptide, and if it's to the intermembrane space, it'll have a second signal peptide, one after the other on the protein sequence. If it's to go to the chloroplast, it'll have a signal peptide, sometimes one or more additional ones to take it to different membranes in the chloroplast, including the thylakoid membrane. And then if it's destined for a peroxisome, it'll have a different signal peptide. Again, a protein will attach and take it to those peroxisomes for delivery. So here are some examples of signal peptides. These are the amino acid sequences. So these would be on the amino end of that growing polypeptide coming out of the ribosome. So if it has this sequence here, then that protein with its attendant ribosome that's translating it at the time are moved to the ER membrane. And then that protein is then threaded into the lumen of the ER membrane. If it's to be retained in the ER, then there's this signal peptide that's actually on the carboxyl end of the protein. So it's a slightly different mechanism there where it's retained in the ER. To go to the mitochondrial matrix, then these are signal peptides, again, on the amino end of that growing polypeptide or protein. This one would take that growing protein to the nuclear membrane and into the nucleus. And then these ones are for to go to the peroxisome, cell surface, or the chloroplast. Then there are these ones in bacteria and archaea. That's really where these signal peptides began. And you think, well, why would that be needed? Well, for archaea, they mainly go to the plasma membrane, but in bacteria, especially for diderms, or gram-negative bacteria with two different membranes, then there's one destined, these proteins are destined for the inner membrane, for the periplasmic space in between the membranes and the outer membranes. So anytime the protein has to wind up in a membrane or going through a membrane, then you would have a signal peptide on the amino end, sometimes the carboxyl end, but more rarely, to take it through, to be recognized by a protein, to take it to those areas, and then to go through a transporter, a channel, to move it through those membranes. So here's a schematic of some different organisms that have varying membranes. So in a gram-positive bacterium with one membrane, a monoderm, If it has no signal peptide, then that protein ends up in the cytosol. If it is to end up on a membrane or going through the membrane, then it needs a signal peptide. These planktomycetes that have multiple membranes, then they have signal peptides that go in through those different membranes, including that nucleus-like organelle, and then other membrane-bound organelles, such as the anammoxysome. Gram-negative diderm bacteria, well, they have two membranes and a periplasm, and so there are signal peptides that are needed to go through each one of those membranes if they traverse any membrane. So for ancient eukaryotes, as the mitochondria and chloroplast are entering those, initially there's no signal peptide to take it to those new membranes, but those are picked up usually by adopting some of the membrane the signal peptides that go through certain membranes in the original alpha proteobacterium or the cyanobacterium. And so initially those are probably weak signals, and then eventually become very strong signals to take those particular proteins into the various compartments of those cells. Here is a general scheme for how these systems work. So in the protein, there would be a signal peptide, and that would be recognized by a signal recognition protein. That would then bind with a receptor on the membrane of the target membrane. And then there would be a switch over to the membrane transport complex. The protein would be then threaded through by its amino end, and there's a signal peptidase on the other side that cuts the signal peptide off. So then you'd have the mature protein inside of that organelle, whichever organelle it happens to be. Here's an artist's rendition of this process. Here's the ribosome, which is translating that protein, and then there's a signal peptide that emerges on the amino end, which is encoded in that mRNA. And then a signal recognition protein attaches to that. That then is recognized on the membrane of the ER in this case by a signal recognition protein receptor. And then there's a switchover using GTP of that amino acid end into a pore protein, which traverses the membrane of the endoplasmic reticulum. Then there's a signal peptidase on the other side. So as this is threaded through that pore, it interacts with the signal peptidase, which clips off that signal peptide. So now that that protein is continuously then translated by the ribosome and until you get to the carboxyl end, and then it folds into its functional form within the ER lumen. So here are those pathways. So if you get transcription here in the nucleus, then the ribosomes attach to those mRNAs and start to translate the proteins. If those have a signal recognition protein attached to that amino end, then those attach to a signal recognition particle, which takes that particular ribosome with its emerging protein to the ER membrane. It's then threaded into the lumen as it's being translated. And then some of those end up going to the Golgi and winding up producing plasma membrane, peroxisomes, and membranes of some of the other organelles, including the outer membrane of the mitochondrion and chloroplast. Then there are those proteins that will wind up going through the mitochondrial membranes or the chloroplast membranes, either 1, 2, or 3 membranes. And so those are first produced on cytosolic ribosomes, those signal peptides then attach specifically either to the outer, the mitochondrial membrane complexes, or the chloroplast membrane complexes, those pore complexes that then shuttle those proteins through those membranes, either 1, 2, or eventually 3 membranes in some cases. Here is a little more detail of those pore complexes. This, with the receptor in the pore complex, is called the translocase of the outer membrane of the mitochondrion. This is translocase of the inner membrane of the mitochondrion, and then you have ones for the chloroplast too, and the C in each case then stands for the chloroplast, either taking it through one membrane into the inner membrane spaces, or for mitochondria through 2 membranes into the mitochondrial matrix, or 2 membranes into the chloroplast stroma. And then there's also another membrane involved in chloroplast, and that's the thylakoid membrane. So there would be another signal peptide to take it through that particular membrane. So here's a composite of some of the things that happen in the mitochondria. There are actually a few others that occur also, but there's one where the signal peptide attaches to a signal recognition protein, and then this signal peptide receptor binds to the signal recognition protein, and then through GTP, as I've shown, it takes it through both the inner and outer membranes in this case, and then there's a signal peptidase on the other side that cuts off the signal peptide, and then you get folding of that protein. This one's a bit more complex in that there are 2 signal peptides, one on the extreme amino end and one in a little further. So this one, the first one takes it through the first 2 membranes, and then the signal peptidase cuts off that first one. And then there's a second signal peptide that takes it through a different transport protein here, and delivers it to the inner membrane space. There are some where the signal peptide is cut off, and then that protein matures, and there are others where the signal peptide actually becomes part of the protein that inserts into the inner membrane. Third type here has a signal peptide on the end and a signal recognition protein there that attaches to the receptor, threads that through the single outer membrane, and then delivers it usually to the inner membrane, sometimes to the inner membrane space, depending on the particular protein. There are some also in the mitochondrion itself, that is, they are encoded and translated in the mitochondrion that have a signal peptide that would take them through that inner membrane into the inner membrane space, or deliver them to the mitochondrial inner membrane itself. So this shows additional detail in a few cases. You have the signal protein or signal peptide, and then this some proteins wind up going to the inner membrane space. Some go through a second inner membrane translocase that threads that protein into the inner membrane. There are others that wind up in the inner membrane space, and then there are some that go through the inner membrane into the matrix, and then becomes a matrix protein acting in the matrix itself. So it could be something in the TCA cycle, for instance. Then there are some that go through a different process here, going through the outer membrane translocase and then being acted on by another membrane-bound complex that then threads the protein into its final conformation, in this case a beta barrel, to become a transporter in through the outer membrane. And of course there's these ones that are encoded within the mitochondrial genome, and then the mRNA winds up on mitochondrial ribosomes, and then you get threading through of some of those through transporters on the inner membrane, and they wind up in the inner membrane space. This figure is a little more complex because it does show more of the different proteins, additional proteins that are involved in this process. So here's a protein here attached to a chaperone, which is going to deliver that protein to the surface proteins, the receptors on the outer membrane of the mitochondrion. And then those are threaded through this transporter, intermembrane transporter, which has a GTP function also. And so some of those are then taken through a second transporter on the inner membrane, or a second channel. Then a signal peptidase using ATP clips off the signal peptide, and the protein then matures from there. And usually it takes other proteins to fold it so that it becomes mature. Another direction that can be taken by a different type of protein, a carrier protein, that is one that winds up in the membrane, has also a chaperone with a signal sequence that is recognized by cell surface receptors, or mitochondrial membrane receptors. That goes through the same transporter, but then attaches to different types of proteins in the intermembrane space, which causes it to bind and be threaded into a protein that is a maturase. That is, it folds a protein into a particular conformation and inserts it into the membrane, in this case the inner membrane of the mitochondrion. There are analogous pathways for import into chloroplasts, in that there are signal peptides on the amino end of those emerging proteins. Those are recognized by different receptors, binding proteins, including some that are labeled here HSP, which are heat shock proteins. So when the plant goes into heat shock mode, different proteins are delivered to the chloroplast to guard against actually the effects of the heat killing the chloroplast. And then there are other ones that are normally used, and these then thread those proteins specifically through the two membranes, the outer and inner membrane, into the chloroplast stroma, and then those are clipped off of those ends by those signal peptidases. And again, as with mitochondria, there are some proteins that wind up in the inner membrane space, and those can either be produced or move into the stroma and then move back out through that inner membrane, or they can be delivered by certain pores and transport systems, translocases that deliver that protein through the outer membrane into the inner membrane space. But then there's a more complex issue of how do you get into the lumen of the thylakoid. And there are several different methods for doing that, but usually it takes a second signal peptide. So the first signal peptide is clipped off, and then that exposes the second signal peptide, which is recognized by proteins that take that protein to the thylakoid membrane for delivery either to the membrane itself or into the lumen of the thylakoid membrane. So here's a summary of some of those pathways. So here's a signal peptide, signal recognition particle, the receptor of that here, and then these translocases embedded in the membrane. So the first one here goes through both of these membranes, and then the signal peptide is clipped off, and that then becomes part of the stroma, or it's threaded into the thylakoid membrane by other proteins. In the second example here, there are two signal peptides. The first one on the amino end takes it through the first two membranes. That's clipped off, exposing the second signal peptide, which then attaches to other proteins that take it to the thylakoid membrane. and through a translocase through there to become part of the thylakoid lumen. And there are two types of transport here. One is passive transport, where it just threads it through. Another requires ATP in order to move that protein through that thylakoid membrane into the lumen. And then there's another type that has two signal peptides here, where one signal peptide takes it through to the stroma, and that first signal peptide gets clipped off, and then that exposes a second signal peptide that takes it through to the intermembrane space, where it becomes part of the protein component in the intermembrane space, or it gets threaded into the inner membrane of the chloroplast. And then there's another type where the signal peptide is recognized by a recognition particle on the outside that takes it through only one of the two membranes, and that also winds up in the intermembrane space. So you can see that these processes are fairly complex and have taken literally billions of years to become this complex, but they're absolutely necessary for movement of those proteins into the different cellular compartments. Another process that's vital to all cells, including bacterial cells, but especially important in multicellular organisms, is cell communication, and it's somewhat related to protein trafficking in that it relies a lot on membrane-bound structures, proteins in the cell membranes of each one of those cells. So there are different types of signaling from cell to cell. One type is where a signaling molecule is secreted, such as adrenaline or something like that, and then it attaches to a receptor on the target cell, and that signals the target cell to go through some process. There's another one there where the cells are together. There's cell-to-cell signaling, and this would be something like, neurons communicating, or just cells recognizing that they're next to another cell so that then they don't go through cell division. So a lot of different cell processes rely on cell-to-cell signaling. So another name for these ones where you get this cell-to-cell, usually close contact signaling, is paracrine signaling, where one molecule one cell is secreting particular molecules to target cells around it, such as T cells signaling other cells, and macrophages signal other cells in the immune response, etc. You also have synaptic signaling in neurons, where they're releasing a neurotransmitter which is picked up by receptors on the other cell, and then that cell starts an action potential or some other process. Another type of signaling is long-distance signaling, and that happens when you release a chemical, a transmitter of some sort, through the bloodstream to a target cell. So this could be something like insulin being secreted by beta cells in the pancreas, and then those insulin molecules are hitting a receptor on a different cell to open up channels for glucose transfer or some other process. So that is endocrine signaling. Here are some proteins and other molecules that are involved in cancer, and we'll talk about these in more detail later in the semester in a later video. But one of these is a growth factor, an epidermal growth factor, and other prostaglandin growth factors, etc. And here's the receptor which interacts with the growth factor itself and then signals inside the cell. Here's another one called RAS, which then stimulates an inhibitor of transduction, and if it's not inhibit, then that signals a transduction to start gene expression in cells that normally aren't supposed to be going through active gene expression at that time, so that can also cause a cancer cell. So again, that involves cell-to-cell communication. And then here's another one that involves cell-to-cell communication. And this is another growth factor. This is a growth inhibitor factor. So most of the time, if a molecule, a ligand is attached there, it will inhibit the growth. And then here's another one. This is attachment of two cells together. So there's cell-to-cell communication between two cells, which normally will inhibit their growth and division. But if something goes wrong in that process, either one of these molecules isn't there, or one of these molecules down here, these beta-catenins and APC, and these are both involved in colon cancers. If that APC doesn't attach to beta-catenin and the receptor, then the beta-catenin, if it's free out there in the cytosol, moves into another process, another signal transduction pathway that increases gene expression of genes that then cause the cell to grow and divide and proliferate, causing a tumor. These are other proteins and other protein complexes that are involved in cancer and signal transduction, that is, transduction of a signal out here into the cell and throughout the cell. So these ones signal BRCA1 and BRCA2 signal DNA repair and apoptosis, which actually increases the concentration of another protein called P53 over here that regulates apoptosis, that is, killing the cell. If there's too much damage in that cell, P53 is signaled, and that will kill the cell. Then there are these cell cycle inhibitors and also cell expression stimulators, which are affected by these outside signals from one cell to another. And then another one over here, BCL2, which is an apoptosis inhibitor, and that also interacts in that signal transduction pathway in normal cells, as well as in damaged cells, and eventually also in cancer cells. And as with protein trafficking, these cell-to-cell interactions turn out to be much more complex than you think they might be. So here's one, which is activation of RAS, which attaches GTP. So there's a growth factor that attaches to a growth factor receptor. When the growth factor is attached, that changes the conformation of that protein. The protein then interacts with a binding protein that binds to an inactive RAS, which has bound to it GDP. So RAS is a part of a class of protein molecules called G proteins because it binds GDP and GTP. So once this binds, that changes the conformation of RAS. It drops the GDP and picks up GTP, and then that activates RAS, which activates other proteins that stimulate the mitogen-activated protein kinase pathway, the MAP kinase pathway. Kinase adds phosphorus to some proteins, activating them. And then that activates transcription and increase of the cell cycle and mitosis, etc. So just the attachment of this growth factor to the outside starts out this whole cascade of things that are part of the signal transduction pathway within the cell. And that stimulates a large number of genes to start transcribing and making the RNAs, and then eventually the proteins to activate that cell to do particular things, including grow and go through mitosis and divide. So this shows a more elaborate picture of what's going on. So here's the ligand out here that attaches the growth factor that attaches to the protein, the receptor, that starts off a cascade of events to activate RAS, which then causes an this MAP kinase pathway to start up, which starts phosphorylating proteins in these different systems, including these ones, Jun, Myc, and others, which are transcription factors that will increase transcription in that nucleus. There's another group over here that attach to a different receptor, and that's the SRC system, which is named after sarcomas, which are connective tissue cancers. And so those also are part of the cell-to-cell communication and some of the things that fail to function properly in tumor cells. And that is not the end of the complexity of the system. So here are 4 receptors shown here and how they interact in their activity. And so you have a number of these actually turning on other processes within the cell. And then altogether, these are stimulating transcription, increase of expression of certain genes, usually to increase the activity of the cell by taking up more sugar or causing the cell to grow, causing the cell to undergo mitosis and divide and other functions. And then to extend the complexity a little more, you can see that all of these on the outside surface, those are cell surface receptors. So those are either recognizing other cells, there's a cell-to-cell interaction here, or they're receiving growth factors, small molecules that are growth factors, things like estrogen and other hormones. And then this one's called wingnut for frizzled, and that is one that was studied in Drosophila and others, but we have that same system in our cells too. So many of these systems are conserved throughout animals and sometimes all the way through protists and plants. Everything that's kind of this reddish orange color, each one of those genes and gene products, when they mutate, are implicated in some types of cancer. So here's P53, right? That's one of them that controls cell death, apoptosis, although there are others down here that also control cell death, such as BCL2 and BAX and things like that. There are other proteins and genes that change gene expression, cell proliferation, apoptosis, and then there are some that recognize DNA damage and other cellular damage. So there's the RAS I was talking about earlier and the SRC I was talking about earlier. So there's an entire signal transduction pathway. That is, the signal starts here or here at the cell surface and is transmitted through a number of different proteins and systems, including this MAP kinase system, to start expression of more genes and usually winding up abnormally turning those on. So that starts out here at the cell surface and moves all the way through. So that's part of cell-to-cell recognition and simply cell communication. Here is a simpler version of that, just categorizing those functions of those proteins into different categories. So here are the cell surface receptors here, as well as the ligands themselves, the growth factors, cytokines, etc. So those are the receptors on the outside of the cell and the signals that are sent by other cells up here. And then you have signal transduction pathways, including, SRC and RAS systems. And then those signal other things to control gene expression, the cell cycle, and DNA repair. And you can see BRCA1 and BRCA2 implicated in many breast cancers as well as other types of cancers. Will signal P53 if there's too much DNA damage, and then cause P53 to signal cell death for that particular cell. So this is just an example to show you how complex these cell communications can become. And this is a case where the breast cells in mammals are being stimulated to produce milk. So here are the milk proteins being formed way out here. But it involves several different types of cells interacting with one another. So you have prolactin receptors and progesterone receptors that will interact to cause a difference in gene expression in these cells. Those then will signal other cells, adjacent cells, to also communicate to then signal another type of cell which has other receptors on it, including estrogen receptors, laminin receptors, and prolactin receptors. Those start out a cascade of gene expression that then cause the proteins, the milk proteins to be formed in those cells, and then those vesicles where they're made are then they fuse with the cell membrane, releasing the milk into the breast tissue for delivery to the young progeny. So we see all these cell communication molecules and receptors and signal transduction and all that in these multicellular organisms, including animals, fungi, amoeba, plants, stromatophiles. But how far back in evolution do you go with these? And the truth is you go back very close to the beginning. So these cell-to-cell communication systems have been developing and evolving for billions of years. And that includes their appearance in unicellular organisms as well. So the unicellular organisms communicate with other cells of their same species. So it's clear in some species, like Caulobacter, that has more than one cell type, that there is cell-to-cell communication, and there's sensory proteins and receptors on the cell surface, because the stalk cells, once they're cemented down to a surface, they stay there. But then they divide off these swarmer cells that have to swim through the water and find a new spot. And once they do, that signals the flagella to come off and then a stalk to be formed. And all of this takes cell communication, cell-to-cell communication of one sort or another. You can often see this in the oceans around the world. And it was noted by sailors a long, long time ago that in certain parts of the ocean, on dark nights, you could see these fluorescent patches. And if you put a rope down into the water or put a paddle in, that fluorescence would increase around that. So there's obviously something going on. And we know now that those are organisms, single-celled organisms usually, communicating with each other, so that at high concentrations they cause this fluorescence, which is caused by a certain protein, often luciferin, that causes a flash of light, and it's then stimulated to be expressed by communication between those cells. So when the cells are very concentrated, more of that stimulating compound are produced or is produced, and that causes those cells to increase the production of those proteins, including things like luciferase, and so it causes them to have more fluorescence than if there's a small concentration of those organisms there. Then they don't form the luciferase, and you don't see the fluorescence. Because of the fluorescence, then people started looking at, what's causing that? And they discovered that these cells were communicating. But this is not the only way that organisms communicate, nor the only response. So it's only an indication of one response that was sort of obvious because things lit up and people could see them. So there are several different types of interactions that can occur. One is up here, and it's called quorum sensing in bacteria. These tidal flashes also occur with dinoflagellates, with different types of algae, with some animals, etc. So, and you get fluorescence in some fish that are caused by primarily bacteria living in them. But here are two different methods of stimulation by cell-to-cell communication. So in some bacteria, they have a gene that will make an enzyme, and from that enzyme it will cleave a compound or produce a compound that is an inducer. Now, the inducer will then be excreted from those cells, and at high concentration, some of that inducer comes into either this cell or an adjacent cell and binds to a receptor, which becomes a transcription factor to turn on expression of a particular gene or set of genes. Another interaction is when there's a receptor on the outside of the cell. Here's a protein that produces a product that then is excreted from the cell. At low concentrations, it doesn't bind with that receptor of this cell or other cells, but at high concentration, it binds to that receptor. That receptor then activates an associated protein with that. That protein then interacts with another protein, which becomes a transcription factor, which then increases the expression of certain target genes as well. And in some cases, those genes can then produce things like luciferin to create light. But they can also, there are a large number of processes besides just creating fluorescence. So here are three more specific examples of those. In this case, there's a gene called LuxI that forms a compound, a small molecule called a lactone. And the lactone at high concentrations will interact with a receptor, so in only high concentrations with this particular organism, so when they're congregating. And that will turn on certain genes, which lead to production of certain gene products. Another one here produces a signal peptide, so it's a small protein, essentially, fewer than 50 amino acids. And that's transported outside of the cell, and it's transported by an ABC transporter. So it's a transporter that uses ATP to transport that signal peptide outside that interacts with a serine kinase receptor. The serine kinase then phosphorylates another molecule that acts with a response receptor, which becomes a transcription factor to increase the expression of specific genes, which could be the Lux proteins, but Lux protein genes, but also many other gene products. And then this one, the Lux is on quite often, and when you're at high concentrations, some of these processes actually inhibit the production of luciferase. So these do sort of the opposite, where you get high concentrations of this particular compound interacting with that receptor causes them to increase expression of LuxU and LuxO, which inhibits production of luciferase and light. This one also over here does similar things, interacts with the same pathway to inhibit, as well as this set of pathways here, where you get production from LuxS of a lactone over here that interacts with a multiple subunit receptor, which interacts with the same pathway to inhibit the production of luciferase. So these ones up here stimulate the production of gene products such as luciferase, and these ones actually stimulate production of inhibitors of the turning on of the luciferase locus. So if you looked at this phylogram again, all of these are multicellular, and you say, well, of course they have to communicate, otherwise you don't get a coherent being, a coherent organism, right? Your skin cells have to be in communication with other cells around them, and your liver cells have to be in communication with cells around them, etc. And that goes all the way down to multicellular organisms, including bacteria. But then these unicellular organisms in white here also have communication systems. So one individual cell is actually communicating with another individual cell in many different forms. And as evolution progressed, then you start with a small number of those type of receptors and interactions in these bacteria and archaea. And as you go into more and more complex organisms containing more complex organelles and then becoming multicellular, you have to have more and more of those systems to coordinate the organelles, and you have to coordinate the cells in their interactions with one another. So this completes the short section on cell communication. I'll talk more about this when I talk about cancer cells, because there's a lot more involved with those. But essentially you have cells communicating with other cells, both at the unicellular and at the multicellular levels. And the first part of this video was about protein trafficking, which is somewhat related in that you are signaling or trafficking, either one, through membranes. So the membranes have to remain intact for these things to happen. And then protein trafficking involves things that are happening within the cell from one organelle to another, or just through one membrane and another membrane to the outside. And cell-to-cell communication, or cell communication in general, is taking one of those signals from the outside and then moving that to another cell, or changing a protein receptor to signal something inside to start signal transduction or some reaction or reactivity within that responding cell. Okay, thanks. That's the end of this video. I will talk to you later.